is the Director of Ultra-High Field Imaging at the Athinoula A Martinos Center for Biomedical Imaging, which is over in Charlestown. He's Assistant in Biomedical Engineering here at Mass General. He is affiliated faculty at the MIT Division of Health Sciences and Technology, and he is Assistant Professor at the Department of Radiology at Harvard Medical School. Let's welcome Jonathan.
Thanks, Sam, for the introduction. Thanks everyone for coming, especially braving the elements to get here tonight. I really appreciate it. It's a great honor to be here tonight. I'm really looking forward to the presentation, looking forward to your questions. So I've been asked to talk tonight about the 25 years of fMRI. I think you know it's the anniversary of the first fMRI experiment. So just to give a little bit of background, so I'm a researcher, an fMRI researcher. I lead a small group of engineers, physicists, and neuroscientists. We're all looking to develop new technologies for fMRI and apply these technologies to better understand the working unit. And so I'll be talking a little bit about some of our work. I'm going to survey a lot of previous work over the last 25 years. I'm going to take this opportunity to editorialize a little bit. I think there's lots of great materials out there on fMRI. I'm going to share my perspective on some of these topics. And again, look forward to your questions. So as I mentioned, the occasion tonight is it's almost 25 years to the day, anniversary of the first published fMRI study led by Jack Belleva, conducted at the Martinos Center, which is over in Charlestown, now known as the Athinoula A Martinos Center for Biomedical Imaging. Here's a picture of Jack, the lead of that study. Jack sadly passed 2 years ago, but he was sort of the inspiration behind this study. He was the one who had the crazy idea to take over the center. He was a neuroscientist who was dedicated to imaging the functional brain. And he sort of went over to the center and basically convinced a center full of physicists and radiologists who were looking at perfusion all across the body, looking at kidneys, heart. He convinced them to shift their focus to the brain in order to measure the brain during all of function. So I think that we owe a lot of debt to Jack for his vision. And again, I think that given the fact that it's this anniversary, we're going to take some opportunity to look back at the 25 years, kind of see where we've been, see a little bit where we are, and kind of take a look at where we're going. So there are going to be 2 themes to this presentation tonight that I'm going to circle back to many times. The first is that functional MRI works by tracking changes in blood flow and changes in blood oxygenation accompanying neural activity. Okay? So fMRI really isn't a measure of direct spiking neural activity per se. It's a measure of blood flow changes and blood oxygenation changes that accompany neural activity. So this is an indirect measure, a proxy to measure neural activity. What we'll see is that it's actually going to be very precise both in space and time. You can get a lot of information looking at changes in blood flow and blood oxygenation in the brain. The second theme is that functional MRI really has become the predominant tool for measuring brain function non-invasively in humans. So this is really the first technique sort of enabling human neuroscience. For the first time with fMRI, we're able to watch the brain as it's functioning. So this has important implications because, again, for the first time, we're able to observe the brain while it's working. There is important ethical considerations, societal implications of this I'd like to discuss a little bit during this presentation. And also, I should point out that we're not just measuring brain function with fMRI, we're really watching the brain in action. And these pictures of brain activity that we do measure and we do generate with fMRI, have some interesting interpretational issues and considerations that we'll talk about in this presentation. So with these 2 themes, I'm going to give a brief overview of what we're going to be talking about this evening. I'll start with, again, because we're celebrating the anniversary of fMRI, that'd be appropriate to give a brief history of functional MRI, just talk a little bit about how fMRI works. And after that, I'd like to talk about some cool, interesting applications of fMRI that we see today. I'd like to just take a little bit of time to talk about the societal impact and ethical questions that surround human neuroimaging. Again, because fMRI is the sort of predominant method for human neuroimaging. I think these topics have to go hand in hand. And I'd like to give a little brief vision of where I think fMRI is going and what the future may hold. Okay, so some of the original concepts of fMRI actually date back to the late 1800s. So Angelo Nasso recorded a bunch of experiments. He was a physiologist, a Italian physiologist who was interested to track changes in blood flow across the human body. And he developed this system, really interesting system, to measure changes in blood pressure within the skull. This is basically a little drum they placed on top of this neurosurgically created bony defect in the skull. Okay, and with this, he was able to observe changes in blood pressure in these patients. Now, as a control, he also measured pressure within the forearm, and he measured the signal changes when the subject was resting quiet, both in the forearm and in the brain. And what you could detect in these signals is very small fluctuations due to changes in blood pressure driven by the cardiac cycle. So basically, you'd be able to see the heartbeat through these traces. What was interesting was that when he looked at these recordings in the brain, when the subject was performing some cognitive tasks, for example, he asked the subject to answer, what is 8 times 12? And when the subject was asked to perform this cognitive task, he noticed an abrupt change in blood pressure around the prefrontal cortex where this apparatus was recording blood pressure. So changes in blood pressure, changes in blood flow that accompanied normal activity. And he saw another peak when the subject responded answering this question, whereas he saw no changes in the blood in the forearm. So this indicated to him that there could be some local changes in blood flow and blood volume attributed to mental calculation, in this case, due to cognitive changes in the subject. So this is the first demonstration that we know of local blood flow changes in response to neuronal activities. Now this experiment was followed up a few years later by Roy and Sherrington. This is a picture of Sir Charles here. They did a similar study. What they did was they kind of drilled holes in the skulls of experimental animals, also measured blood pressure within the brain, measured changes in the cortical diameter. And what they did was they cut some of the nerves, feeding into those brain regions, and kind of electrically stimulated those nerves, causing electrical activity in the regions that they were measuring. And what they found is after this kind of weak electrical stimulation, they saw very large changes in brain diameter, very large changes in blood pressure accompanying these electrical changes at the neurons. So this is the first sort of direct evidence, the first direct observation that the brain blood flow regulation could be affected by local neuronal activity. What they found was very presciently that the brain possesses an intrinsic mechanism by which its vascular supply can be varied locally and in correspondence with local variations in functional activity. So this may not be very surprising. The fact is the brain is like a muscle. When neurons start to fire, they have more energetic demands, they need more oxygen. So it makes sense that the brain would divert fresh blood, fresh oxygenated blood to regions of the brain that require. One of the main questions, though, today in fMRI is still unanswered, is how precise is this blood flow regulation? To what extent can the brain really deliver blood exactly to the regions that need it? This is an open question that we're currently investigating. I think what we've learned is that the more we look, the more we find that the brain is actually very good at diverting blood exactly to where it's needed. But the point is today we still don't know exactly how fine-scale the blood flow regulation is in the brain. But this is the first sort of demonstration of this. Now, to leap forward about a century, the first imaging technique that was able to be performed to measure function in human brains and living human brains was actually positron emission tomography. This really caught hold in the 80s. In positron emission tomography, what one does is labels some molecule, like glucose or oxygen, with some radioactive tracer. And what happens is, over time, that tracer, that molecule, will build up in whatever brain region that's absorbing either glucose or oxygen. So it builds up over time, and one can image the pattern of radioactive tracer in the brain using positron emission tomography to know which parts of the brain were absorbing that chemical. Okay, so here's an example of subject scanned at rest. You can see the distribution of blood flow across the subject's brain. And the same subject scanned about a few minutes after they performed a very simple motor task, in this case a finger tapping, asked to kind of tap their fingers, which activated their motor cortex. You can see a local increase, a focal increase in blood flow to that region as detected with PET imaging. Another cool example of PET in action, this is a case in which subjects were presented with a flashing checkerboard stimulus, which activated the visual cortex, which is in the back of the head. So what you can see is that by tagging oxygen or by tagging glucose, we can estimate after a few minutes of presenting the stimulus to the subject, we can tell what parts of the brain are responding by having increased blood flow, increased glucose utilization. Okay, so PET is a really great example of a functional neuroimaging technique. There are some disadvantages, however, to PET. One is that, as I mentioned with PET, we're looking at the buildup of these radioactive tracers in parts of the brain that accumulate the chemical of interest. So of course, a few scans are okay, but this is something you don't want to do over and over and over again because there's some radioactivity involved. Another issue with PET is that it only gives us one sort of snapshot of brain activity. We're able to see over the period of minutes, which brain region was sort of active, sustained over that period of time. It's only sort of one snapshot of what the brain had been doing for a period of minutes. It doesn't give us the dynamic information that we really want in order to track brain activity over time. So inspired by these PET studies, I think a lot of people in the MR community were kind of interested to see if MRI could be used to track brain activity. And as I mentioned, Jack was sort of dead set on looking to see if MRI could be used in this regard. And a lot of his colleagues at then the NMR Center were developing techniques to measure changes in perfusion in different parts of the body. And Jack thought that these perfusion-based techniques could be used to track brain function in humans. So here's an example of the first fMRI experiment. So just to walk you through this, so this experiment, what Jack used was a contrast agent, gadolinium, which is actually used routinely every day in the MR clinic. This is a contrast agent that's injected into the blood and has some magnetic properties to it. Okay? So the way this experiment works is, because gadolinium has magnetic properties, whenever it passes through the brain, it causes a local decrease in the MR image intensity signal. So what we can see here is that Jack, set up the subject, asked them to view a kind of blank screen, and gave them an injection of gadolinium into the arm, intravenously. The gadolinium went into the heart, got pumped into the head, and he observed the signal within the visual cortex. What he observed is as the gadolinium within the blood passed through the visual cortex, through the blood in the visual cortex, it caused a local decrease in the MR signal, as it was passing through on its way back into the heart and through the circulatory system. Okay? So a local decrease, which was when the subject wasn't really doing anything in particular, observing a kind of neutral gray screen. Then he repeated the experiment. He gave the subject another injection, but this time he presented with them a visual stimulation. Okay, so the same thing happened. The gadolinium went in through the heart, got pumped into the brain, passed through the visual cortex. In voxels, or volume pixels, that are sampling within the subject's visual cortex, he observed a local transient decrease in signal as the gadolinium passed through the tissue. But this time, the signal decrease was greater. He saw a larger decrease when the subjects were observing this flash-injected stimulation. And what does that mean? What it means here is that because there's local increase of blood flow during the neuronal activity within the visual cortex, what's going on is that the vessels were distended, they were enlarged, so more gadolinium could pass through those vessels because their diameters were bigger, causing a higher increase or an increase in the concentration of gadolinium of this magnetic contrast agent, and therefore the MR signal went down even further compared to the baseline state. Okay, so that's how this experiment worked. So what they did was they compared the signal intensities between the on conditions when the subject was looking at the stimulus and the off conditions. They compared the minimum intensity between these two cases and looked across the brain at which voxels were statistically, using some statistical analysis, had some difference between their intensity during the on state and their intensity during the off state. And this here is a map shown in kind of this heat scale of those voxels that had a significant difference between these two states. So by measuring this change in the signal intensity as the gadolinium was passing through the brain, Jack was able to determine which voxels were responding to this stimulation. Again, indicating that these voxels had larger amounts of gadolinium in their voxels because the vessels were distended during the sustained neuronal activity. Okay, so this is the first fMRI experiment. So there's some drawbacks to this experiment that maybe you might guess. So one is, kind of like the PET, we're administering a contrast agent. Gadolinium is safe, it's used every day in the clinic, but this is not something you want to repeat over and over again. Having that much gadolinium in your body might be unhealthy. But similar to the PET experiment, this only really allows us to kind of compare two states. It allows us to compare the state in which the subject is looking at a blank gray screen and the state in which the subject is looking at this flashing chartboard. It doesn't really enable us to look at dynamic information. So again, this is the first demonstration of fMRI applied at humans, but it really isn't the way we use fMRI today. It inspired new techniques that were looking for other mechanisms other than gadolinium, other than this contrast agent, to look for changes in blood flow and oxygenation in the brain. And one such mechanism is to note the fact that there already is iron in the blood. There is some mechanism through which we can get local changes in magnetic fields, just without using contrast agent, but just from the blood itself. And that's because red blood cells red blood cells have hemoglobin. Hemoglobin, just remember, is the principal oxygen carrier in the blood. Now, when hemoglobin is oxygenated, basically it has no magnetic signal. It has the same sort of magnetic properties as water. We really can't distinguish oxygenated hemoglobin from water, and therefore it's kind of invisible in MRI scans. What happens is when the oxygen is delivered to the tissue and is stripped off of the hemoglobin turns into its deoxygenated state, and the iron in the hemoglobin is all of a sudden exposed. It's no longer shielded by the oxygen, it's exposed, and this deoxygenated hemoglobin becomes paramagnetic. It becomes magnetic, it causes local field disturbances in the MRI machine, which we can use to track where there are local increases in the concentration of deoxyhemoglobin. This is the principle that lies behind current technique of functional imaging, was commonly called the blood oxygen level dependent technique. What we're able to do here is we're able to track changes in the concentration of deoxyhemoglobin following neuronal activity because deoxyhemoglobin is magnetic. And basically, you can think of it as we have little magnets in the blood, that are only really exposed when their oxygen was turned off. That's the principle behind fMRI today. Now this technique, this bulb technique, was first demonstrated in vivo by Seiji Ogata's team. So what he did here was he imaged an experimental animal, kind of breathing normal room air. He could detect the venous vasculature in this animal, where there's a high concentration of deoxygenated hemoglobin. You can see these sort of dark vessels in this animal's brain. But then what he did was he imaged the same animal, but he put the animal in a room full of 100% oxygen. Okay, so this kind of saturated all of the deoxyhemoglobin, kind of removed all of the deoxyhemoglobin, replacing it with oxygenated hemoglobin, which has no signal. And they imaged the same animal again, and you can see all of those dark vessels disappeared. Okay, so this is the first sort of in vivo demonstration of this bold concept applied with MRI. So the idea here is that there's some MR signal that could be made sensitive to a local concentration of deoxyhemoglobin. Can we use this to measure brain activity? The answer is yes. Here's an example. This is Ken Kwong's first bold fMRI study. This is Ken modeling the goggles that he used to deliver the stimuli to these subjects. So what Ken did was he sensitized his MR experiment to this bold contrast. You can see that when the stimulus was off, the image is pretty much flat. When he turned the stimulus on, all of a sudden, he saw a big signal change in the back of the head. Again, the visual cortex here in the back of the head. And they saw the signal modulation following the visual stimulation. Okay. Now, many other groups have since replicated this. I think the Ogawa group, also inspired by Jack's work, by these early PET studies, also realized that this bold signal it could also be used for tracking brain activity. But Ken was able to demonstrate this first. This is actually a movie. I'm not sure where I found this. I got this a few years back. This is a movie of the first demonstration of bold fMRI. This was presented at the 1991 Society for Magnetic Resonance in Medicine meeting. This was presented in one of the plenary lectures. And it's pretty cool because what you can see here is this is a movie of brain activity. So it's kind of upside down. The back of the head is at the top of the image. So you can see that as the subject is viewing this visual stimulation, you see this bright increase, there it is, in signal as the change of deoxyhemoglobin accompanying brain activation is modulated. Okay, so this is the first demonstration of fMRI using, in this case, not gadolinium as in Jack's study. In this case, we're using an endogenous contrast agent. A contrast agent that's already in the body is taking advantage of the fact that oxygen kind of modulates the magnetic properties of hemoglobin. Okay. So I think what in the 90s, I think people were very excited about this technique. We saw that as we deliver visual stimulation, we can see changes in visual marks in the brain. I think people are very excited about this technique. There was some skepticism in the field, frankly, in terms of how do we interpret this signal? This is blood flow. I mean, is blood flow really faithfully representing neural activity? And I think a real landmark study was conducted in 2001 by Nikos Logothetis. So what Nikos did was he developed a very sophisticated system for simultaneously recording neuronal activity, electrical activity, and bold fMRI. Now, it's a little hard to see in this plot, so I kind of drew over it myself there. So the curves to the left are indicating different aspects of electrical activity, spiking. And this plot in red is demonstrating the bold response that immediately follows that spiking activity. So you can see the bold response is a little slow compared to the neural activity. That's because it takes vessels a little bit of time to open up and for the blood oxygenation to change. But the point is that what Nico saw was very tight correlation between electrical activity and these bold signals throughout the brain. And what's interesting is that, again, we had a suspicion, or the field had a suspicion that fMRI was, faithfully representing neural activity, but really wasn't until this study, I think, that a lot of neuroscientists came on board. In fact, I think for years after this paper came out, virtually every paper I read on fMRI began with a paragraph saying, fMRI is a faithful measure of neural activity, and they cited Nikos. So I think this was a real kind of pivotal moment in fMRI where the neuroscience community, who had been skeptical, kind of started to come around to this as a technique for measuring brain function non-invasively. So now one thing I do want to point out though is that fMRI doesn't give us a picture of brain activity. Nikos showed us that it does represent neural activity, but the data itself is pretty noisy. It's not exactly a veridical picture of brain function or mental processes. So this is an image. This is the data as it comes off our MRI scanner. So the point is, every MRI scanner today, any kind of modern scanner can do fMRI. We don't need a special scanner. But these are the data that we get off of these scanners after conducting an fMRI experiment. This experiment, the subject, as you can probably tell, was looking at a flashing checkerboard and performing a finger counting task. I don't know about you, it's very hard to see any fluctuations in this data whatsoever, especially probably from your setting. If you squint, you can see some little fluctuations, but very little. This is very far from a photograph of brain activation. So one thing we can do is we can subtract the mean image, kind of like what Ken did in his movies, get a better sense for how these images are changing with this stimulation. So you can see here in the back of the head again, there's some fluctuations in the intensity that seem to be kind of visually driven with this checkered border pattern. We can also see some changes in the motor cortex that seem to be driven by this finger tapping task. But this is, again, far from a picture of mental activity. We see a lot of fluctuations throughout the brain, even in regions that shouldn't be responding to these stimuli or tasks. So the question is, what's going on here? There's lots of noise in the data, lots of changes in the image intensity, but not really driven by our experimental manipulations. And the name of the game in fMRI analysis is to identify those fluctuations that are being driven by our stimulus or task, kind of separate them from those fluctuations that we don't understand, fluctuations that seem to be due to noise. So if you look at this voxel on the back of the brain, you can see that its intensity is roughly following the on-off pattern of our visual stimulation. We turn the stimulus on and off and on and off, and we seem to be modulating those voxels in the back of the brain correspondingly. And again, with fMRI analysis, what we do is we take this sort of pattern of activation that we're inducing using our stimulation, kind of an idealized neural response. We kind of correlate this, we compare this sort of idealized representation of the neural response, looking for voxels whose intensities match the pattern of the stimulation. So this is how we identify which voxels in the brain are responding to our stimulus or task. But as you can see, again, there's many fluctuations in the brain we really can't account for using this analysis. So I think what's really important in experimental design is to really make sure that we compare two conditions that really allow us to identify which parts of the brain are responding to the stimulation of interest. So this is a really nice example, I think, from Nancy Kammerscher's lab, in which she presented subjects with pictures of faces and pictures of objects. She found there's a cluster of voxels, cluster of locations in the brain, where the voxels are responding more to the pictures of faces than they are responding to pictures of objects. But it still wasn't clear, is it really faces that these voxels are responding to? Maybe it's some other aspect of the image that Nancy wasn't thinking of. So she tried a little bit harder. She said, okay, well what if I take these pictures of the faces and I cut them up, kind of scramble them around, so that they no longer look like faces, but they have the same sort of distribution of bright voxels and dark voxels, or I should say pixels, such that she can account for this potential reason why these voxels are responding to the original stimulation. What she found was, again, you compare these intact faces to the scrambled faces, you saw that the voxels within this region were responding much more strongly to the intact faces than to the scrambled faces, indicating, again, giving us more confidence that these voxels really are specific to stimuli consisting of human faces. Now, by the way, this study was quite remarkable. I think that this has been replicated many, many times. This is one of the most robust findings, I think, in fMRI, this sort of face area and the fusiform gyres. It also gave rise to this sort of theory, this modular theory of brain organization. It's kind of astounding that there's one brain region that's sort of devoted to processing human faces. Are there other brain regions that are similarly specialized? Is the brain consisting of many of these modules that each of which correspond to a different processing mechanism? It's still unclear, but it's an interesting theory that is still being tested today. This notion of functional specialization, however, goes back many, many years. This is a kind of anatomical atlas generated by Corbini and Brodmann, again in the early 1900s. What he did was he took some brain samples and put them under the microscope and observed that there were striking differences in the microanatomy of different brain regions that he was able to use to sort of discriminate or identify different brain regions across the cortex. So here's a map that he discovered. It's still largely accurate. I think we're still basing a lot of our studies based on this anatomical parcellation or anatomical division of the brain. Well, what's interesting is that a lot of these anatomical borders that Brodmann identified by placing specimens under microscopes match up pretty well with functional borders. We know that the motor cortex, for example, lines up pretty well with this Brodmann area, kind of astounding. Visual cortex is another great example. We see that there's a lot of structure-function correlation in the brain. There's some deviations from this. And we also know that different functions are kind of clustered in different anatomical regions. Memory, audition, speech. Many of these functions are specialized and do appear in different parts of the brain. And a lot of what fMRI really seeks to do is answer the where question. Most of fMRI studies today present the subject with some kind of cognitive task or some stimulation and try to identify which part of the brain is responsible for processing that task. Most studies are concerned with identifying these functional specializations. fMRI is not only used for cognitive science and basic neuroscience. These mapping techniques are also used in brain surgery. Actually, what's amazing is that I found this from the Aetna webpage. Aetna is a medical insurance company. They actually will reimburse doctors for performing fMRI to use to plan brain surgeries. The idea here is that when the surgeons are trying to decide how much tissue to remove, say there's a brain tumor or some sort of lesion, they want to make sure that they remove the lesion, but leave the eloquent or leave the living tissue surrounding that tumor, especially in cases where the tissue close to the tumor is really important for that subject's well-being. For example, if that tissue is responsible for language production or motor control, it's really important for the well-being of that subject to retain those tissues. So fMRI is actually used quite a lot in pre-surgical evaluation to figure out exactly which parts of the brain are responding to these stimuli, again, in order to resect the smallest region possible and leave those parts of the brain that are important for those subjects' well-being. Here's a kind of common example of using fMRI to plan brain surgery. So in this case, subjects were asked to perform a kind of silent word generation task, to imagine words in their head, and they plotted out the activation within the language network. And mostly, language is either left dominant or right dominant. I think most of the time the surgeons just want to know which hemisphere is responsible for language within that subject. But here again, they can map out eloquent cortex, living cortex that is still involved in speech production in order to make sure to not remove that during the surgery. Another use of fMRI in terms of mapping out is to map out organization within individual cortical areas. One principle that we've looked at quite a lot is the principle of retinotopy. Retinotopy is this principle that's shared by many different animals in which nearby neurons in the visual cortex possess receptive fields that are nearby in the visual field. So just unpack this for a second. Most of the neurons in the visual cortex are kind of looking at some spotlight in the visual field. And if stimulation occurs in that spotlight, that neuron will start to fire. What's interesting is that nearby neurons in the cortex are looking at nearby spotlights. So if we extrapolate this out, what it means is that there's a topographic map of the visual representation of the visual input on the cortical surface. So what this means is that if you could picture the activation of my neurons right now, it would resemble the scene that I'm looking at in front of me. So this is sort of captured by this cartoon. So we say that there's a topographic map of the visual field laid out on the cortical surface. What's also interesting is that there's a variable magnification. Notice that you tend to have more detail in the center of your visual field compared to the periphery. And that's actually represented in this mapping. There are more neurons devoted to representing the center of the visual field than to the periphery. That's actually captured quite nicely by this cartoon. We see more cortex devoted to representing the woman's face compared to her arms, which are out in the visual periphery. So we thought this was cool, and we thought this would be a nice kind of way to understand whether we can map out organization within individual cortical areas and get a sense for whether fMRI could be used to map out fine-scale organization within the brain. Again, this is a technique that's based on tracking changes in blood flow. How precise is it? So we wanted to exploit this mapping. We designed a stimulus to create a pattern of activation on the visual cortex, in this case in the shape of this letter M. So we computed, based on this known mapping, how we would have to, the shape of the visual stimulus that we have to present to that subject in order to elicit this pattern of activation. So here's what our kind of warped M looks like. Again, we had to flip the M because the retina, of course, inverts vertically the visual input. This is our condition A, condition B. So here's what the subject looked at, kind of sped up by 5 times. This is our stimulation that we used in order to elicit this pattern of activation on the cortex. Well, the problem is it's a little bit hard to see this pattern of activation across the cortex in the sort of native images of fMRI. The map is kind of crumpled and kind of tucked into these folds of the cortical folding pattern. Kind of hard to see the organization. One tool that we have in fMRI to kind of look at these organizations across the cortex is to model the cortical surface using MRI data. As I mentioned, in our MRI experiments, we're conducting fMRI using the same scanners we use for MRI. So there are some techniques that we have that kind of accentuate anatomical contrast. So this is an image that's been designed to highlight contrast between cortical gray matter and white matter. So the gray matter is the part of this thin tissue on the surface of the brain where all the sensory processing and a lot of higher level cognitive functions take place. And so in these data, we can distinguish the cortical gray matter from the subjacent white matter. We can draw a boundary between the gray matter and white matter, shown here in yellow, and from that we can build a surface reconstruction, a model of the subject's cortical surface. And once we have this model, we can better examine the patterns of activity across the cortex rather than the folded image. So here's an example. This is that same activation I showed you in the previous slide, but now presented on the cortical surface. So you can see in the original representation, it's a little bit hard to see the M. It's kind of buried within that fold. But if we sort of inflate the surface like a balloon, literally like inflate the mesh, just to kind of visualize the pattern of activation there, you can see that once we've done this, we can better detect in this the spatial pattern of this M activation within the human visual cortex. Now this is a very small chunk of cortex. I think it's about like half of a business card or so. The point is we see a lot of detail in these maps. This is not bad for a technique that's based on tracking blood flow. I think that this evidence and many other studies have shown us, surprisingly, that the fMRI signal can be very spatially precise, much more than I think we believed even just a few years ago. Here's another fun example of this. We also know that there's these topographic maps in many sensory and motor systems. We know that there's a similar sort of topographic map of finger representations within the precentral gyrus. So here's an example. This rainbow map is showing you representations of the little finger, the ring finger, middle finger, index finger, thumb, all down these precentral gyrus. So there's individual representations for each of these fingers embedded on the cortical surface that we can see with these surface-based analyses. It's pretty cool. We can also see within individual digits, we can see progressions of representations of different pieces of the finger, again, in the other direction. So the point is here that with fMRI, even though we're looking at changes in blood flow, we're able to see very fine-scale organizations within individual cortical areas of the neuronal representations of body parts and sensory inputs. Now, here's an example. Neuroplasticity is another cool application of fMRI. This is something that we cannot assess using behavioral tasks. This is an example, I think, that Bruce Rosen mentioned in the previous talk. I thought it was really cool, so I wanted to include it here. So in this case, these subjects, normal-sighted subjects, were given some language processing task, and they were put in an fMRI machine during this task, and the typical language network lit up. Not too surprising, but then the group put in some congenitally blind adults who had been blind since birth, and gave them the same language processing tasks. What they saw was some of the same language regions were activated, but they also saw a lot of activation within the visual cortex. Okay, so again, the visual cortex, which is ostensibly silent in the blind adults who have been blind since birth, it seemed like the visual cortex had been recruited to participate in some of this language processing. So this is a nice example of cross-modal or cross-sensory recruitment that we can detect with fMRI. So again, this not only does tell us that language processing in the visual cortex can be possible in congenitally blind subjects, but the same group showed a kind of similar study in which they simply blindfolded normal-sighted subjects for a couple of days. And after about 5 days in this case, they gave these subjects a similar language processing task. And even after being blindfolded for just a few days, they also saw some interesting activations within these subjects' visual cortices. So it seems that this sort of cross-modal recruitment, this neuronal plasticity in adults, can even take place after a short period of time. So these are some interesting facets of neuronal organization and plasticity that we can measure with fMRI. Now, I would be remiss if I didn't talk a little bit about resting state fMRI and the default mode network. This has really represented, I think, a paradigm shift in fMRI in the past 10 years or so. So just to explain this briefly, so this is a map of a constellation of cortical areas that appear in many different studies, both in PET imaging and in fMRI, to kind of quiet down during most task activation studies. These just conspicuously had lower activation in many different studies. And so when the subject was put in the scanner and asked to kind of lay in a kind of resting state and a default state, these areas tended to quiet down. What's interesting, if you put subjects in the scanner and ask them to do nothing in particular, just sort of rest comfortably, what happens is neurons never stop firing. Neurons are always, firing away a little bit, and they're never completely silent. They have some level of spontaneous activation, and this sort of spontaneous, looks almost random activation of the neurons causes like random-looking fluctuations in the volt signals as well. And so what we can see is that these signals look random, but what's interesting is that if you compare these signals across different parts of the brain, what we see oftentimes is that even though they look random, these signals can be strongly correlated between very kind of distant brain regions. So it seems that because they're random, the only way in which we could see these sort of correlated fluctuations is if these regions are talking to one another. There's some communication, there's some potential anatomical connection from one region to the next that's correlating these seemingly random fluctuations. So what's interesting is that just by looking at these patterns of fluctuations in the resting brain, we can try to infer which regions are talking to one another, which regions are connected to one another. This is known as resting state functional connectivity, trying to look to see which regions are talking to one another from this sort of seemingly random resting state data. What's interesting is that if people looked at these same constellation of areas within the default mode network, what they found was that there's very strong levels, very high levels of correlations between these nodes. So this sort of default mode, this constellation of regions that were quieter during task activations, seem to be strongly correlated as well, and therefore constitute a network. So the default mode network is actually one of the most robust findings, I think, in fMRI. It's something that we've seen over and over again. It's really hard to not see the default mode network. It's used quite a bit to compare activations across different patient populations and groups. People have observed that the default mode network can be an interesting biomarker to distinguish between different populations of subjects. Now, the default mode network is not the only whole brain network in the brain. There's many others. Several of them have been identified and named. What's interesting, however, is that any given brain region may be participating in multiple of these kind of non-overlapping networks, just as many of you are probably participating in multiple sort of non-overlapping social networks, such as your friends, your family, high school classmates. You participate in multiple networks at the same time, and so are these brain regions. What's interesting is that brain scientists have been looking at these networks that they're deriving from resting state fMRI data. What they're finding is that the structures of these networks actually resemble, in many cases, sort of organic networks we see very often in biology. And a lot of these networks also resemble some of the social networks that we see in different platforms. So, and one point that I want to make here is that across the centuries, it seems to me that people always like to build analogies between the brain and the current technology of the day. I think a couple centuries ago, people thought that the brain was like a clock, and they built analogies between brains and clocks, because that was the technology of the day. Later on, the brain became the steam engine, because that was the technology of the day. People built analogies between brains and steam engines to try to understand brains better. In the 80s and 90s especially, I think people built analogies between brain and computers. Computers were the technology of the day, and we tried to understand the brain in terms of understanding our understanding of computers, computer science. Today, it seems that the brain is a social network. A lot of network scientists are exploring the brain and and we have learned a lot about the brain through examining its structure, examining the network properties of the brain using resting state functional connectivity. Another interesting example of fMRI in action is so-called brain decoding. So this is a cool example. This is from Jack Gallant's lab at Berkeley. So what Jack did here was he presented subjects with these little movie clips, and over again, scanned them for hundreds of hours, measured their brain activity, sent them home, brought them back a couple days later, brought in some new people, and showed them the same clips, and measured their brain activity again. What they tried to do from the second session, they measured their brain activity, they tried to guess from the measured brain activity what the subjects were watching at any given moment. Let's start that again. So the images on the left are the movies that the subjects were being shown, and the images on the right were Jack's best guess as to what the subject had been looking at based on their fMRI data. So not quite mind reading, but definitely Jack is able to tell what you're watching. This is known as brain decoding. This is a new technique. Now you can see that the images on the right, aren't perfect. but I think that's pretty impressive. I think that this is also, by the way, this is 5 years old now. I know that Jack today is implementing a lot of new technologies that he's deriving from the math that's being developed for deep learning and big data in order to improve the accuracy of this brain decoding mechanism. I think this is really encouraging, and it tells us again that there's a lot of information in these fMRI signals that can tell us a lot about what the brain is doing in real time. Yes. I thought I'd also be remiss if I didn't talk about the dead salmon fMRI. This is a very popular study that's part of fMRI culture, I think. This is a fun study. Actually, I attended this poster at the Society for Neuroscience meeting in 2009. I really enjoyed this. So what these investigators did was they took a dead salmon, they put the salmon in the scanner, they presented stimuli, they asked the salmon questions, they performed a neural experiment, acquired fMRI data, and analyzed the data, and lo and behold, they were able to see activation in certain parts of the nervous system. Now, to be clear, we do not expect there to be any activation in this dead salmon. These are false positives, these are errors. And I think when the media got hold of this, they had a field day, of course, saying that fMRI, how can fMRI be valid if it's claiming to see, if these people see activation, even in a dead salmon. And what's interesting about this study, though, is that it was an educational poster that was meant to be fun. They were trying to make a point here, making an argument for so-called multiple comparisons correction. This is a step in fMRI analysis that's done so automatically that I think students are even tired of hearing about it. It's done automatically within the software, fMRI software. And so I think this poster was meant to remind trainees that multiple comparisons correction is important. And I think that they made this point very nicely. Again, I think that because it was a little tongue-in-cheek, I think that the media ran away with this. But again, I think it's an important point, and therefore I want to talk a little bit about what is this multiple comparisons correction? How is it that we're seeing activations in dead salmon? Well, one analogy that I like to think of is that it's basically due to the fact that there are random fluctuations in our fMRI data that every once in a while look like activation. So the way I like to think about this is that consider an fMRI experiment, there's some random noise, there's some statistical properties to this data that's kind of akin to like you flipping a coin many, many times. So say you've got a single voxel, you're measuring 100 time points in your fMRI scan. So you can think of like the fluctuations due to noise within this voxel as being many, many flips of this coin. Okay, sort of random, statistically random fluctuations. So the question is, if there's some noise processing in the brain, how likely is it that you're going to flip a coin in such a way as to look like your activation? How likely is it that you're going to flip the coin such that you get 5 heads in a row, then 5 tails in a row, then 5 heads in a row, then 5 tails in a row, then 5 heads in a row? I mean, it's very unlikely. Right? If you flip a coin again, it still looks pretty random. We're not getting this pattern. It's extremely unlikely to get this exact pattern of heads and tails. But fMRI can easily have 1 million voxels in any one measurement. So even if there's 1 in a million chance to flip this exact pattern of heads and tails, if you flip a coin enough times, especially since there's so many voxels of these brains, eventually, just by chance, you're going to get a match. And so this is the so-called multiple comparisons question. If we're comparing our expected activation against noisy data multiple times, eventually, by chance, again, this is just coincidence, this isn't activation, eventually, by chance, we're going to get the pattern that looks like 5 heads in a row, 5 tails in a row, 5 heads in a row, 5 tails in a row. Just by virtue of the fact there's so many voxels in the brain. And this is the point that these investigators are trying to make. Don't forget, trainees, to do your multiple comparisons correction. It's quite straightforward to estimate how likely it is to get these erroneous or spurious activations, given how many voxels you have in your data. So I like this study. I think it was a fun study, but I think it was maybe misinterpreted by some of the media. It's certainly worth mentioning. Another study, this is another kind of example of a potential fallacy, a potential issue with fMRI. This is a study that was published in the New York Times. This is a study in which they tried to investigate love for one's iPhone. So I think what they did here, I don't know the details exactly, I think that they put subjects in the scanner and showed them pictures of iPhones, measured brain activity, and what they saw was a lot of activation in the insula. And the authors concluded, therefore, because the insula is involved with strong emotions and with love, clearly these people love their iPhones. Even if you think you're an Android lover, no, your brain tells you that you love the iPhone, and therefore you have to accept that. Now, the problem with this is that this is an overinterpretation of the data. This is known as the reverse inference fallacy. The idea here is that if you see some activation in the insula, it's really hard to know what the subject was experiencing or feeling or thinking during that time. Just because the insula is involved with emotional regulation doesn't mean you can infer exactly the emotional state of the subject. Actually, what's amazing is that there was a couple of blog posts that came out immediately after this, demonstrating that the insula is activated in almost 30% of all fMRI studies. The insula is involved with lots and lots of different things, lots of different emotions, positive, negative, what have you. So the fact is that just because it's involved, in some studies it's involved with love, doesn't mean we can infer from seeing in any one study, like the study looking at iPhones, we can't infer that just because this area is activating, that means the subjects are